Welcome Bio 107 students. In this video, we're going to learn how we can use a microscope to estimate the size of common biological specimens. To illustrate this concept, here are some examples of commonly studied biological specimens. For example, we have insects. This is a little, I think it's a dust mite or a small flea. We have human cells, such as a neuronal cell. This is a dendritic cell that is part of our nervous system. Here's an example, a little cartoon of a bacteria. Here we have a plant cell, and of course we have a virus here as well. So ask yourself if you know how big these items are. What's the biggest? What's the smallest? Can you estimate their size with your fingers? Well, of course not, right? They're too small. Maybe the flea, right? Maybe you could kind of put your fingers as close as possible together and kind of get an idea of how big that flea might be. But can you describe the flea in terms of centimeters or millimeters? Is a bacteria bigger or smaller than a virus? Which is bigger, a plant cell or an animal cell? These are questions that a microscope can answer. So let's see how we can measure using a microscope. So before we get into how a microscope specifically is used, let's remember that we are now scientists. Whether you like it or not, you're a scientist, and we're going to use the metric system. So let's quickly review and just kind of get a feel for what the metric system means in terms of microscopy. Remember that the metric unit of length is the meter, and a meter is a little bit over 3 feet. So if you think about what a yard is about the same as a meter. So we're going to use the meter as our unit of distance, but of course when we're looking at small things, a meter is way too big. So we need to make that meter smaller by adding prefixes to the meter. So a commonly used smaller unit of measure is of course a centimeter. And if you have a ruler in your home, I would recommend pause the video quick and go grab it. Maybe dig around in your kid's art box or something. If you have a ruler, chances are that ruler on one side of it has inches and on the other side of it it's going to have centimeters. So if you probably remember this side as the side that you never used when you were growing up. So this side is the centimeters, and you can see that about 2.5 centimeters is equal to 1 inch, right? So you can kind of in your mind remember or recall, and if you have that ruler in front of you, you can see what an inch is. And here I just included some coins, right? Some common American coins here as a reference to give you again an everyday sense of what a centimeter might be. We can see here that a penny is about 2 centimeters in diameter. So the width of 1 penny is really close to 2 centimeters across. But centimeters is still too big. Remember, we're talking about microscopy, things that we really couldn't even see with our own eyes, with the naked eye. So centimeters is still too big. So let's go a little bit smaller. Remember that a centimeter is 1/100th of a meter. And we want to go a little bit smaller to a millimeter or a millimeter. And a millimeter is 1/1000th of a meter. So 10 millimeters is the same as 1 centimeter. So again, if you have your little ruler here, or you can look at this one, of course, those little tiny hash marks that divide a centimeter into 10 equal intervals, each one of these little hash marks is a millimeter. So this is a millimeter. Again, too big. This is a millimeter is just starting to get to the size where we are getting useful for microscopy. We want to go one step further, and we want to go to the level of the micrometer. A micrometer is 1/1000th of a millimeter. So think about this tiny little space here, these little hash marks. If we were to take that little tiny hash mark and divide it into 1,000 equal pieces or equal lengths, that would be a micrometer. So a micrometer, 1,000 micrometers is equal to 1 millimeter. And a common term for a micrometer, some people call it a micron. It's just easier to say. It's less syllables, I guess. So you might, if you're watching a video or reading about this, you might hear it be referred to as a micron. And the symbol for a micrometer is this weird looking kind of U symbol. And this symbol is the Greek symbol for the letter M. It's called mu. And this is the symbol that represents micro in the metric system. So whenever you see this kind of weird U and then M, we're referring to microns or micrometers. And again, these are tiny, tiny, tiny little links. We for sure cannot see this with the naked eye. And this is really where we're at with a light microscope. A light microscope or a bright field microscope is good at illustrating objects that are in the micrometer or micron range. So how do we do this, right? We can't, a ruler's too big. We can't put a ruler in a microscope, or can we? We can, it turns out. So what we do in microscopy is we put a little lens on the ocular eyepiece. Remember, the ocular piece is the part at the top that kind of looks like binoculars that you look through. And in that eyepiece, in the ocular lens, we're going to put a little overlay of a piece of glass that has a little tiny ruler kind of etched into the glass. And what this does when you look through, so imagine you're looking through the microscope right now, and in your field of vision, you see some specimen that's on the stage that you're looking at. And what happens is you have a nice little ruler that is overlaid on top of or through that image that you're looking at. And in this way, you can kind of count the hash marks in the length that would represent, in this case, the length or the width of whatever this this looks like, some type of protist or protozoan that you may be looking at. And so in this way, we can estimate the size. Now, of course, the question that you're asking is, what is the units, right? What does this ruler represent? Is it inches? Well, of course not, right? Is it microns? Is it micrometers? Well, we don't know. And so this is kind of the important point where many biology students kind of get tripped up. The size of the hash marks depends on the magnification that you're looking through and the size of the ocular micrometer itself. In other words, it varies from microscope to microscope. So anytime you get a new microscope, you need to calibrate your ocular micrometer so you know what this ruler actually represents. Let's look at an example here. So here, for example, if we were actually in lab doing this with our compound microscopes, we have already calibrated our microscopes. We know that when we use the microscopes that are in the Bio 107 labs, the scanning objective has a calibration of 25 micrometers for every ocular unit. So an ocular unit is a fancy way of saying this little hash mark here. Right? So every little mark is an ocular unit. So for the scanning objective lens, that means that each little hash mark represents 25 micrometers. If we move up, if we go up in magnification to the low power objective, then each hash mark represents 10 micrometers per ocular unit. And finally, if we use our most powerful high power objective lens, we see that each hash mark represents 2.5 micrometers for every little hash mark. In other words, if we wanted to know the actual size of this little protozoan guy here, and we wanted to know how big it truly is in real life, in actuality, we need to know two things. We need to know the calibration of the ocular micrometer, so which lens we're using and what its calibration is. And then we also need to count the width of this actual object. If we look at this, it was 5, 10, 15, 20, 25, 30, 35. So it's approximately 35 ocular units across. Right, so we know the width, but we don't know, in this particular case, we don't know which objective lens we're using, so we can't calculate this particular example. But let's do an example where we can calculate it. Also, just if you, a note to the listener here, you probably want to pause the video here and jot this chart down. It's going to be very handy for you when you do the quiz. So if you haven't done that yet, go ahead and write that down quick. Okay, so now let's look at an example. We want to measure a red dot. Super important that we get we estimate this red dot in size. And so we're going to start. Remember, we always start on the lowest power objective lens, which is that shouldn't say 4x, that should say 40. Not sure why it says 40. I'm gonna fix it really quick. There we go. Okay, so we're going to start on the 40x. Remember, our scanning objective lens has a total magnification of 40x. And my calibration tells me that that means I have 25 micrometers for every ocular unit I'm counting. So at the 40x with scanning objective lens, I can see that this red dot spans roughly 1 ocular unit. So I know the width of this is 1 ocular unit. So pause the video and see if you can determine the actual width of the object. So hopefully you recall that the width of the object times the calibration of the micrometer will give us the actual width. In other words, 1 times 25 will give us the actual width of the object. So in this case, our math is fairly straightforward. 1 times 25 gives us 25 microns across. So in other words, if each of these ocular units span 25 micrometers, then that means that this red dot is 25 microns across. Now, let's move up in magnification. Let's go to the low power objective lens. Remember, the low power objective lens is stronger than the scanning objective. So now we're magnifying this red dot 100 times. And notice that now we can actually see the red dot a little bit better. And we can see that it's not so much a dot as it is an oval, right? It's not a perfect circle. It's actually a little bit more of an oval. And we can see that this oval spans 1 to about 2.5 ocular units across. So if we write this into our graph here, 2.5, and we do our calculation, what is the actual width of our red dot? Well, 2 things to think about. 1, does the red dot change depending on our magnification? Does the actual width of our red dot change depending on our magnification? No. Because, I mean, think about this. If we have something on the stage of our microscope and we're looking at it through our lens, and we change the objective lens, that doesn't change the length of the actual size of the red dot, right? Imagine if you're 6 foot tall and someone's looking at you close up and someone looks at you from far away, you get bigger and smaller depending on where someone is looking at you. But you're still 6 foot tall, right? Your height and your weight and your size doesn't change, even though you might look bigger or smaller depending on how far away someone is when they're looking at you. So it's the same concept here in microscopy. Even though we increased our size, increased the power of our objective lens, and our dot looks bigger to us, the actual dot didn't change in size. So notice that you still get 25 micrometers across, or 25 microns across. All right, let's try it now with our high power, right? We're increasing the power of our magnification. We're now at 400 times magnification. And our dot got even bigger, and we can even more clearly see that it's an oval now. And we notice that it's roughly 10 ocular units across. And of course, by now you know that the actual width didn't change. It's still 25 microns across. And we also can double check our math. 10 times 2.5, of course, gives us 25. So there's a little walkthrough of how you can calculate the magnification at each of the different the 3 objective lenses that we use in 107. So now I'm going to challenge you and make you do the calculations yourself for the objects that we talked about in the beginning of this video. So we first have a mite, this little mite or flea. I'm not sure. I'm not an entomologist. I'm not sure what it is. I just grabbed the image off Google. And we're going to look at this mite, this little flea, under the scanning objective lens. And see if you can, first pause the video and see if you can estimate how many ocular units it is across, and then see if you agree with me. Okay, so I'm going to estimate here's 1 to 5, 5 to 10, and 10 to 15. So to me, it's about 15 ocular units across. Is that what? Oh, good. I put 15 in. Great. So we know it's 15, and at the scanning objective magnification, we know that each ocular unit is 25 microns across. So now you need to do that calculation to determine how long the dust mite or the flea's body is. And then write that number down because it will be in the quiz. All right, so now let's try it. Well, first of all, let's go back here. So we're going to do the plant cell next. And let me ask you, do you think a plant cell, an individual cell that makes up a plant, of course they vary in size. They can be, there's a range of sizes. But do we think a plant cell is bigger or smaller than a flea? Well, hopefully I mean, if we think about this, cells have to be, for the most part, smaller than I mean, this is a flea is a multicellular organism, right? It's made up of hundreds, well, actually probably even thousands, right? Tens of thousands of cells. So a cell is much, much smaller than an organism like an insect. So if we were to look at a plant cell under a 40x scanning objective lens, it would be way too small, right? If I made it too small, you couldn't see it, but I don't know if you can see that tiny little green dot right there, right? It's too small. We only can use the 40x scanning objective to kind of find our sample to focus. Remember, adjust that coarse focus. So now let's zoom in and use the low power. Now we can kind of start to see it. But let's go up one more and adjust our coarse focus. And now we can see that plant cell really nicely, and we can estimate its size by counting across the ocular units. So go ahead and count the ocular units and see if you agree with me. So which row are we going to use this in? Remember, we're looking at the high power. So we're going to insert our number here in that bottom row. And I counted 9. Let's just agree with 9. I think it might be 8 or 9, but let's just agree with 9 so that we all do the math in the same way. So if this is 9 ocular units across at the high power lens, then what is the actual size of this plant cell? Again, do that calculation. Pause the video if you need to. Do that calculation and make sure you write it down so that you can take the quiz. All right, let's do our next example. Here we have a neuronal cell, or neuron cell, and we're observing it under the high power lens. So we're all the way at the bottom. We're at 400 times magnification. And if you haven't taken anatomy yet, this part, dendritic cells or dendrites have a kind of two parts to them. They have a main body, which is called the soma, and then they have this really long tail that's called the, oh my goodness, the axon, I think it's called the axon. And these, the axon can vary in length quite a bit. In fact, we have nerve cells, we have dendritic cells that look, dendrites that look like this that are, I think, about 1 meter long, like very, very long cells, and they can also be very short. But the soma, the main body that contains the nucleus, and this is where all the protein synthesis takes place, this is a little bit more consistent in size. So let's estimate the width of the soma. Again, pause the video, see if you can count across. And I think I counted 13. Yeah, great. 13 ocular units across. So go ahead, write down your calculations and make sure you make note of that for the quiz. And our final example are bacteria and viruses. So if we're under the high power objective lens, remember high power is 400 times, we can maybe just start to see bacteria. Bacteria are really, really small, much smaller than animal cells like a plant cell, or much smaller than eukaryotic cells like plant cells or animal cells like a neuron or a red blood cell or any eukaryotic cell that you can imagine. Prokaryotes, bacteria, are really, really small. In fact, too small really to be effectively seen using the high power lens. We would need a more powerful microscope that contains an oil immersion lens in order to kind of reliably look at and estimate the size of bacteria. Now, even using the oil immersion lens, which I believe oil immersion lens are 1000x, so we have 1000 times magnification where we can start to see bacteria. Even using that oil immersion lens, we couldn't see viruses. Viruses are much, much smaller even than bacteria, even when compared to bacteria. We would need an electron microscope to determine or to be able to see viruses and to be able to estimate their size. So we would need a microscope that's much more powerful than a bright field microscope that we use in most science labs in order to see viruses reliably. So I wanted to end this lecture just by giving you an idea of the range of sizes. Again, think about, a typical human is between 5 and 8 feet tall. I'm not sure why they think that we're 1 meter tall. That would mean that we're only about 3 feet tall, which is not true. We're, typically between 5 feet. So we're usually between, I would say, 1.5 meters and 2 meters. I would say most 90% of humans fall in that range from 1.5 to 2 meters. And then everyone, you can kind of imagine what a chicken egg looks like. That's about between, again, it varies, 10 to a 10 millimeters would be a really small egg. Yeah, that's way too small. I would say that an egg is probably about 4 centimeters in diameter across. But I wanted to get down here. Notice that these are the things that we can see with the naked eye. Whereas these smaller objects are the objects that we would see in a light microscope, because a light microscope will magnify an item anywhere from 10 times to about 1,000 times for a good oil immersion lens. And we can see that using a light microscope. But when we get really small, when we start to talk about viruses and big macromolecules like proteins and lipids and DNA, we need an electron microscope in order to do that. So in this video, you learned the basics of an ocular micrometer and how to estimate size of the object that you're looking at in a microscope using that ocular micrometer. Good luck on the quiz.